Okay, so for those of you who don't know me, which is no one in this room, you all know me, but my name is Joselynn Wallace. I'm a genomics data scientist with the Computational Biology Core, which is part of the CCV, the Center for Computation and Visualization. So today I'm going to talk a little bit about some of my favorite genome annotation resources in R. Mostly we're going to talk about Annotation Hub, Biomart, and quite a bit of genomic features as well. Okay, so first we can load all our libraries. And I guess I should start by telling you a little bit about how this workshop is going to be set up to run. So we're going to run everything on RStudio on Singularity. And you can request resources that look like this. You'll only need an hour, 4 cores, maybe 8 gigs of memory. The path to the Singularity container is in the chat, but I'll paste it again. And then the path for the R executable, you should just give it the full path to your Scratch directory, because it's where some like temp files will get written. And then for additional data path, you should give it that folder, because then you'll be able to actually open the notebook. Once all that's filled out, then you can just hit launch. And then you should see, how do I see? Here we go. Once you do that, you'll be taken to this page, and when it's ready, you'll see this button that says connect to RStudio server. And when you do that, yes.
I have a question, please.
Yes, hello.
Can we put our path to our Scratch?
Yeah.
Do we have to use like the real path, or is like /users/stay/Scratch okay?
I don't know. Do you want to try it and see if that works? Let me know if you run into issue. Okay. So once that's all done and you're in the session, you'll probably have to go to File, Open File, and that's where you want to give it that path to here. To that additional data path, and then hit open. And then this is the notebook that you want. Paul and Eric, I feel like you guys are my true guinea pigs. Are you good to go if you're following along? Okay, great. Okay, so once you open the notebook, you can run this first code chunk, which just loads in a bunch of libraries. And the first package we're going to talk about is Biomart. So Biomart makes it really easy to query these public repositories. We can use it to query Ensemble for our annotations, and we can look for these housekeeping genes, which are. Do you want to join the Zoom and I'll make you host, or someone? Because otherwise I'm gonna have to keep letting people in, which is fine, but let's see, admit. And then people who, I don't know if I can copy my previous messages. Okay, so if you're just joining, you go there. Thanks, Paul. Make host. Great. And people on the Zoom, feel free to unmute and just ask questions if you have them. I'm happy to answer them, and we should have plenty of time. Is there a way to like hide this, I wonder? Yeah, there we go. Okay. So yeah, we're going to use BioMart first, and we're going to use it to look at these housekeeping genes that I found in some publications where they were looking for some stable express housekeeping genes in the TCGA data. So it's like RNA-seq data from different cancer cell lines. And I put the links to the 2 papers that I used here. If you want to check those out, you can do that. So we loaded BioMart already up here. So the list of the housekeeping genes that I picked, we're going to name them in this vector first. And then we can use this list marts function to see which biomarts are available. I almost always just use this ensemble mart ensemble. You might, when we run this function, you might get a thing about how saying the ensemble site is unresponsive. Whenever I get that, I always just use the US West mirror. It hasn't steered me wrong yet. So basically you're going to say, okay, we want to use ensemble, and the biomart we want to use is this one, the ensemble mart ensemble. And I'm just specifying the US West mirror ahead of time. Okay, so, oops. If we type ensemble in the console, it'll tell us that it's, we're using this ensemble biomart database, but there's no data set that is selected. So you can see what your options are for like the available data sets for this mart if you use the list data sets function. There's going to be a lot of them. There's like 200 or something. So we can narrow it down a little bit and only look for human data. So I'm going to use that. I'm going to do that by using the search data sets function. I'm going to specify the mart that we just used, and the pattern is H sapiens, so just human data. And we can see that they have some human genes for GRCH38, which is just one of the human genome versions. And so we can kind of put it all together to create a biomart object. Again, if you get errors about how biomart is unresponsive, you can just use the US West mirror. Okay, and then I think if we, okay, so now it's telling us that we're using this H sapiens gene ensemble data set. Okay, so in a little bit, we're going to use this get BM function. And that's how you actually query biomart. And that's kind of like I feel like this function is sort of the real meat and potatoes of the biomart package. But it'll take the following arguments. You have to give it some attributes. So basically tell it what attributes you want to retrieve. You can use filters to tell it that you want to use these filters when you do the query. And then values would be like the values that you want those filters to take. And mart is obviously the biomart object that you want to use. To see, you can see a list of the attributes using this list attributes function. We're just going to look at the first 5 because there's quite a few. There's about 3,000 of them. But you can see that it has things like the different versions of the gene IDs, the transcript IDs, the protein IDs. And there's other, like, you can get different other types of gene IDs. So like if you want to look at the gene symbol, you can pull out that. They even have stuff like if you work with something that's not human and you want to look at like what the homologs are in human, they have all that information in there too. It's pretty massive. But for the purposes of this workshop, all we really care about is ensemble gene ID and HGNC symbol. So you can use that list. You can use this list filters function to see kind of what your filtering options are. We're going to limit it again to the first 5, but you can kind of play around with this and see what your options are. There's a lot of them. Okay, and then we can kind of pull all that together to build our get BM query. So we're saying that our attributes that we want are ensemble gene ID and HGNC symbol. The filters we're going to use are HGNC symbol because that's what those housekeeping genes that I picked were all in that HGNC symbol format. And then we're telling them that the values we want to filter are those housekeeping genes and the Mars ensemble. And so you can see that for those housekeeping genes that we put in. We have the gene symbol column, and then we also have this ensemble gene ID column. Does anyone have any questions about that? What about in the chat? Nope. Okay.
Yes.
So now you have a handle on how to use Biomart and build Biomart queries. The other one I wanted to talk about is Annotation Hub. And you can use it to do something similar to what we just did with Biomart. And many of the data types that they have stored in Annotation Hub are based on these Annotation DB object classes. So that includes things like org DBs, the TX DBs, and lots of others. And it's nice because that means that a lot of them will have a lot of these different data types or data classes will have like very similar functions that you use to interact with them. So the first thing we can do is use this Annotation Hub function to actually connect to Annotation Hub. And it might ask you, you might get a message about creating a cache folder when you run this command. And I always just say, yes, make a cache for me. Yes. Hello, sir.
Are you sharing your screen, Joselynn?
Am I sharing my screen? Oh, perhaps not. Good catch. Sorry, y'all. Host has disabled screen sharing.
Take blame on that.
Thanks, Paul.
Try now.
Sorry, everyone. I didn't realize that I was not sharing. Okay. How is this? Thank you, Catherine. Okay, so now you can actually see what I'm doing. We are running Annotation Hub. Okay, so after we connect to Annotation Hub, we can see that there's a ton of stuff in here. There's all these different data providers, species, data classes, all sorts of fun stuff. So we can use these like $ accessions to kind of get a handle on what we've got available here. So here I'm just saying, okay, show me all the unique data providers and tell me how many there are. So there's 52 data providers. And that includes things like UCSC, Ensemble. There's other, like I think even some of these like organism specific, like the Drosophila specific annotation resources are in there as well. And then we can also look at the types of data classes. Again, tons of those. There's like the genomic ranges objects. There's just data frames, TXDBs, all kinds of good stuff. One of the options you'll see in there at the end is these org-DB options, which are really nice. These are organism-specific genome-wide annotation files, and we can use them to do a lot of things. And we can use them to do kind of something similar we did with Biomart, where we map between different gene ID types. And you know, if you look on Biomart, you'll see a bunch of these org-DBs, and the names of them will look something like this. Where the first two letters are kind of a 2-letter abbreviation of the organism. ID is an abbreviation kind of describing the central identifier. So like if it's, you know, so you can have like Entres gene IDs, etc. e.g. for Entres gene ID. Okay, so we can see what our options are if we want to look at like the human org DBs in Annotation Hub. We can do that by building these Annotation Hub queries. And it's sort of nice because you can use just this pattern argument to specify these sort of, it's kind of nice that it's a little bit unstructured. So if you know that you want to look at Homo sapiens or sea urchin or whatever, you would just need to put that in that pattern argument. And then it'll give you back so you can see here that there is an org DB for Homo sapiens that uses Entres IDs. So we can grab that first org DB here. That's what this function is doing. Okay, so then we can see, okay, what kinds of information can we retrieve from the org DB? We can use that key types function to find out. So there's all these like accession number, alias, ensemble. entrez, ontology, gene symbol. So one potential use case is the one we just did with Biomart, where you want to use it to convert between different gene ID types. And so one way you can do that is using the select function. And select will retrieve all the data as a data frame. And so the columns will show what kinds of data can be returned for the annotation DB object. The keys will show you what kinds of keys are in the database, and key types will let you discover what kinds of key types can be passed, obviously. And you can view the columns and the key types, and usually they are the same, but not always, just to keep things interesting. So if we look at the columns, and then we can look at the key types too. See if that works. Yeah, you can see that they are the same in this particular case, but don't be lulled into a false sense of security, because this is often not true. Okay, so we can try selecting just those housekeeping genes in the org DB, and we can try to get their ensemble and entrez gene IDs. So we're using this select function. We're saying that our keys are the housekeeping genes, and that we want back these columns, ensemble and entrez, and that our key types in our keys file are gene symbols. So there's the gene symbol, the ensemble, and the entrez gene IDs. And this is a nice example, because you get back these one-to-one mappings between the keys and the columns. But that is not always the case. So if we, for example, wanted to look at the Go terms associated with each of these genes, we can specify that here in our columns argument and say we want ensemble and Go. The key type is symbol. And so you can see that sort of each given gene belongs to multiple Go terms, and each Go term is on a new row. And this might not be your ideal outcome. Let me see if there's a question in the chat. Oh, so all of this is on Oscar on Open on Demand. So you're not going to have like the GPFS data files on your local machine. All this is happening on here on Open on Demand. If you just go to ood.ccv.brown.edu, you should be able to go to RStudio on Singularity. And I put all of the options here in the Slack channel. Okay, so we just talked about what might happen if you're the columns that you want to have a one-to-one relationship with the key types that you've given. So that might not be your ideal outcome. And so one option is you can use this map IDs function. It's sort of similar in that it uses keys and key types, but one important difference is that here the argument is column, not columns, and you can only return one column type. By default, it'll return only the first match, which again is probably not what you actually want. You probably really want all of the Go terms. In that case, you can specify this multi-vals argument. And there's a couple different ways that you can sort of deal with these one-to-many matches. Again, the default is first, where it'll just give you the first value. You can also tell it to give you a list. You can say filter, like if there's you only want certain Go terms, you can put those in there in your filter. You can tell it to just get rid of anything where there's multiple matches. Character list. You can also write your own function about how you want it to deal with multiple matches. Maybe you want it to concatenate everything and have it be comma separated or something. So you could write a little function to have it do that and stick that in there as your argument. We can just say here, we want them to all be a list. And then so you'll get back something like this, where each each gene is an element, and then each gene has this like list of Go terms associated with it, which is probably what you want, not just the first Go term. Okay. One of the other options in Annotation Hub that's like a pretty handy choice is the TXDBs. These are also based on these annotation DB objects, but they're more they're more focused, they're more transcript oriented. So basically it contains feature IDs for transcripts and genes. So TXDB objects can be used to link those gene IDs and transcript IDs. So again, we can query our Annotation Hub. We can specify which record we want to use. Here we're just using this, which one are you using? These known genes. So just like we did with the orgdb, we can look at the key types. We can use select in a similar way. We can look at the gene ID and the transcripts and transcript IDs. We can also use map IDs to tell it to give us a list if there's multiple matches. And then if you want to look at all the transcripts, you can use this transcripts function to see a list of all the transcripts in the TXDB. And you'll get back a gRanges object with the genomic location of each transcript, as well as its like transcript ID and transcript name. If you want to know more about gRanges or genomic ranges objects, you can go here to check out the documentation. I feel like it's I feel like that's one that a lot of bioconductor packages use, so it's worth looking at a little. You can also look at sort of transcripts grouped by the genes that they're associated with using this transcripts by function. So here you can see all the transcripts in their genes. Again, there's similar functions to look at exons, coding sequences, introns by transcripts, all kinds of fun stuff. One function that I think is extremely handy, and I feel like this is one that this is a question we get in office hours all the time, which is like, okay, I have these two data sets that I want to look at, but one of them is UCSC and one of them is ensemble. And so you'll have different, if you get your data from UCSC, chromosome 1 will be called CHR1 versus from ensemble, it'll be just 1, which is kind of annoying. So you can use this seq level style function, which is exported from the genome info DB package to kind of figure out what the current seq levels are of your object and to kind of rename them according to a given style. So we can look at our TXDB and see that we are currently in the UCSC format, right? That's from seq level style. And then if we look at seq info, you can see that what I just explained about the UCSC data, where they name their chromosomes in this way. And we can say, okay, let's convert to NCBI. Just using the seq level style function. And so then you can see that now, instead of CHR1, it's just 1. And instead of CHR, I'm assuming this is like unmapped or something. Now they're in this other NCBI like a format. And if you want to know more about like what specific styles are supported for your organism of interest, you can use this genome styles function. And so it says there's like NCBI, UCSC, DBSNP, ensemble, and is that all? Is there one more? Yeah, that's it. And then you can convert sort of between easily. You can go back to UCSC if you want, and then you can even filter them using the seq levels function to just say, okay, I only want to look at chromosome 15 if you really want to. I will take any questions that you have. That's all I have for this workshop. there any more questions in the chat? Let's see. Oh, great to be. Yeah, sorry. And I think the DSI folks recorded this. So if you want to go back, you can watch it, and I will. This code isn't on GitHub yet, but I'll push it up there before we leave, and I'll put the GitHub link on the Slack channel. Any questions? I love it, Paul. Yes.
So yeah, no, this is great because I always use just like the raw databases, like not like this like really nice way of querying things like through annotation code.
Yeah.
So and I just have a quick question about like
Mhm.
should I say this? Maybe you covered this already, but like you know, it seems like annotation cloud they combine a lot of different sources and you show like the different styles you can even switch, right? Like the US SCS or the
Mhm.
other ones NCBI. I'm just wondering if when you import from these multiple sources,
Mhm.
do you ever run into issues of like, you know, having to duplicate genes, or like say like, you know, one database says, you know, there's like, you know,
Oh,
two
right.
genes, but
Yes.
they're really the same, like,
Yeah.
or one, like, sorry, now I'm tacking on like, or if you have to filter out,
Yeah.
like, say, like one annotation set that's like, you know, really the canonical set, but then there's maybe pseudo genes that's like, you know,
Yeah, and sometimes, and I sort of glossed over it, but sometimes you can have a situation where there's like multiple, the gene annotation like conversions aren't always one-to-one, right? I showed an example where they were one-to-one, but sometimes they're not. I usually get around that by just like collapsing all the duplicates into like comma separated values so that I know. I mean, it sort of depends on like what your use case is, but that's usually what I do just because I'm apprehensive about just like picking one randomly, obviously. So I usually just do some sort of, I don't know if I'm actually answering your question.
no, you are, and but I guess the
Hmm.
follow-up question, you know, like with all
Yeah.
these IDs, like, you know, ensemble, Trez, I'm curious, what is like if there is at all like the primary key for all these IDs, or there is none, it's just like all these independent.
I feel like you can't go wrong with just having the like ranges of the object. That's really the key is like, oh yeah, but I don't think there's like a,
major distributor uses a different algorithm.
Yeah.
The other part even, funny, the question I had, Jocelyn, we looked into this, but so when you change the seq levels.
Yep.
UCSC to ensemble, for example, or NCBI, do they
like, do you lose anything?
How do the unmapped and unplaced and random contigs get figured out, because they're different across different sets.
Yeah.
And like, you know,
Mhm.
the main ones are,
Right, like here.
like, so they're not always.
Let's see.
It's not always the one-to-one. I mean, go out with like look at the ensemble or the other one. I mean, the lengths are different. So.
Why? Well, I should name. I'll name these different things, maybe.
What together with every night sun.
Is this? I don't remember what the other one was.
Yeah,
Let's just do this. Oh wait, this is after I did the filtering. I see, that's why.
I want to know, like, I think.
Well, the lengths of these actually look. I mean, that's only the last 5, but.
Okay.
But yeah, they're not always gonna agree because they use different different algorithms to do like the gene prediction and the structural annotation and stuff like that. So. But I feel like using seq level style is probably safer than just saying, oh yeah, prepend everything in this column with CHR and then we're done. Because there's like, you know, obviously even here in this case of these like unmapped things, like the names of them, it isn't a matter of just slapping a CHR on the front and being done with it. If only. Yes, Paul.
Suppose I wanted to use annotation hub on
Yes.
Stronghold, for example.
Ooh.
Do you know if they provide like an offline mirror or something that we could host internally or something like that?
I would be surprised if they didn't have some sort of like standalone database option that you could
Yeah.
for at least some things for sure. Do you have
I
thoughts?
think they, given, have an option to install local only kind of thing. So I think it's like a certificate.
Yeah,
So you basically, for example, download it, then
right. You just download the SQLite database or whatever.
we'll have to. Or,
And you can point it at that SQLite database.
well, I mean, if you install it and you're doing our install on this system, then put it down to the packages. You use the library or use the original file, the library and it finds automatically for that. So, one of the installation points,
Is that relevant? Is that like something that is of interest to Stronghold users, you think?
Yeah, I'm always curious what, I mean, the question hasn't been raised yet, but.
Right.
maybe just because they can still.
Great.
you have an open thing to expand that or whatever on my connection.
We don't have bioconductor, but we run our own mirror program. Yeah,
Oh, that's fun.
yeah, Pi too.
Cool. Okay,
Thanks, Justin.
thank you.